Hi, everyone. Welcome back to the showcase stage. My name is Rachel, and I'm a technical product manager here at Oxford Nanopore Technologies. And while some of you joined us yesterday while we were talking about human genomics, today we're going to focus on another amazing application of Oxford Nanopore Technology, and that is single-cell sequencing. Single-cell research has exploded recently in popularity, and we're excited to be at the forefront of that research. That is because we offer unique advantages with our technology, which we'll get into in a minute. And I'm hoping to tell you, as well as my colleague Carly, why our platform is great for single-cell research. So one obvious reason why we're so well suited for single-cell research is that with Oxford Nanopore long reads, you miss much less compared to short reads. The current short-read transcriptomics methods miss 80% of available data. So despite your best efforts, you could be missing entire isoforms, splice variants, and much more. Just as one example, our long reads allow for capturing entire transcripts in a single read, showing you exactly what's going on without having to piece together many short reads. Our technology also allows you to answer some of the questions that you see on this slide, and I think that's pretty neat. Some of these questions, such as, what if I could go back to old samples in my freezer to discover more? Or could I find alternative splicing that could play a key role in my research? So many things to consider that you can answer with nanopore technology. So you might be thinking that this must be complicated to set up, but I assure you, it is not. We currently provide multiple end-to-end protocols to assist you throughout the process, all the way from library preparation through to sequencing and to analysis. On our Nanopore community, you can find both a 3' and 5' protocol that is compatible with the 10x Genomics Chromium system. You'll hear a bit more about this from Carly very soon. So it is my pleasure to introduce Carly Tyre to the stage to talk a bit more in depth about single-cell sequencing on our platform and present some data to you all. Carly is a development scientist that works on single-cell, and I'm sure by the end of her talk, you'll be even more confident that Nanopore is really great for single-cell research. So please welcome Carly to the stage.
Thank you. Thank you, Rachel. As Rachel has highlighted, single-cell data provides deeper information for your biological question. Nanopore long-read sequencing of full-length transcripts provides a more complex view of isoform expression, alternative splicing, and fusion events in individual cells. So I'd like to start by highlighting our end-to-end workflow. We'll start with sample and library preparation. So our Nanopore platform is compatible with the NextGem and GemX 3' and 5' gene expression kits, as well as the Visium spatial gene expression kit. For prepping a Nanopore library, you'll need about 10 nanograms of cDNA to get started, and then you'll need our 2 Nanopore kits, the LSK114 ligation sequencing kit, and then as well as our PCR expansion kit, and we'll explain more about that later. So for Nanopore sequencing, it will be 1 Promethean flow cell per 10x genomics sample, and then each Promethean flow cell will be generating approximately 80 million of cell-assigned reads per Promethean flow cell. And then on the data analysis side, we have a really easy workflow called single cell that's hosted on our EPI2ME platform. And that should again generate QC metrics, gene and isoform count matrices, tagged BAMs, and UMAP plots. So I'd like to highlight that our library prep is very fast and scalable. So what we're starting with here is 1 sample will be in 1 tube, and then 1 tube will be 1 Promethean library. So it's going to be really easy to do multiple samples at the same time. And it's 3 hours from cDNA to sequencing, so it's very fast. So our Promethean P24 device can run 24 flow cells at a single time. So they're all in parallel. And if you're generating 80 million cell-assigned reads per each flow cell, and you're running the Promethean at full capacity, you're generating 1.9 billion cell-assigned reads at a single time. That's a lot of data. And if you're running 24 or more samples, our workflow is about 70% faster than other long-read platforms. And we also have a really nice ability to walk away from this actual preparation. So it's 90% of this actual workflow time is Nanopore sequencing itself. So I'd like to highlight how the 10x system works. You'll be providing the 10x chromium with single cells or nuclei, and then the chromium machine is going to take it through the chemical reactions to generate cDNA. So what you're going to want to consider here is your cell type, your cell size, viability, and cell count. You'll also want to decrease cell aggregates and decrease any dead cells, as well as any inhibitors of the reverse transcription reaction. So what you're looking for here is to start your sample with about 90% viability, and anything less than that will just be increasing the noise in your final data set. So you can decrease noise here by using wide bore pipette tips to help decrease any lysis prior to this preparation. So this works through droplet encapsulation. So in your droplets, you're going to have your cells, your reagents, your barcodes, and it's going to go through this process using this adapter here on a gel bead. So you're going to have a PCR primer, which is the read one sequence, the cell barcode, the UMI, and then the poly-T region that's going to prime off the poly-A tail of your transcript. The UMIs are going to help decrease PCR bias and then also allow you to generate consensus sequences from your RNA molecules. So at the end of this process, all of the RNA in a single cell will receive a single barcode, and all of the transcripts within that cell will receive a unique UMI. So at the end of this process, your 10x preparation, you'll end with full-length barcoded cDNA. So we have two preparations depending on which 10x prep you're doing. So we have the 3' protocol and the 5' protocol. In here, the 3' protocol and 5' protocol are going to just be capturing different ends of that transcript that's been prepped through the chromium machine. So you're looking for both of these preparations, where the 3' protocol is going to be using a biotin to just enrich for those full-length transcripts. Both of these preparations do, however, use the poly-T to prime off the poly-A tail, and then they also use the template switching oligo, and that's just going to reverse transcribe and then generate double-stranded cDNA. So I'd like to highlight our 3' protocol. So this is when you're going to be prepping from the 3' 10x preparation. So what the reads you're looking for here are going to be containing a read one portion and then a template switching oligo portion. There's two main steps to this protocol. The first is the biotin pull down and then the PCR off the biotin bead. So you're going to actually, these are primers that are user generated, so you're going to order these in advance to get started with this protocol. You're going to be using a biotin tag on your forward primer, which is going to contain an ONT and partial read one primer, and then you're going to be using a reverse primer of the partial TSO and then our ONT primer. So once you have that PCR product, you're going to do a streptravidin pull down, and this is actually where you're going to be enriching for those full-length transcripts. So we're going to be depleting any background reads. And then you're going to do a PCR off the streptravidin bead. And this is going to be using our PRM primer. So those are the primers that are going to be supplied in the PCR expansion kit, and the tube will actually say PRM. And then once you have that PCR product, you're going to do a spray clean, and then you're going to be ready for just following our standard ligation sequencing preparation. So this is where you're going to take the PCR product, you're going to enter our end prep step, and that's just to de-tail our cDNA reads. And then you're going to be starting this with about 200 femtomoles, so you'll have some extra product to go back to if necessary. And then once you have your end prep step, you're going to be doing the ligation of our sequencing adapters, and those are provided in the LSK114 kit. The tube is going to say LA on it for ligation adapters. And then once you have that ready, you're ready to load your flow cell. So this is just going to be our standard Prometheon flow cell preparation. So you'll flush the flow cell, load your sample, and at this stage, you'll be loading about 50 to 100 femtomoles of cDNA, which you'll be calculating based off the expected read lengths of your fragments. So with the 3' protocol, we're typically expecting about 900 base pairs in our read lengths. So at that rate, you'll be adding about 65 nanograms of your final library onto the flow cell. So for the 5' protocol, the template switching oligo is in a different location. So it's actually adjacent now to the barcode and the UMI. So we're not going to be getting a lot of off-priming. So we're not going to be doing the biotin pull down for this specific protocol. So you'll just be starting with the PCR. These are also user supplied, so they will need to be ordered in advance before you can get started with this protocol. They're similar design to what we talked about before, so an ONT primer with the partial read one, and then a partial non-poly DT with the ONT primer. So you'll do your 8-cycle PCR, a spray cleanup, and then you'll take this final PCR product directly into NPrep and ligation sequencing that we just talked about. So then you'll prep it just the same, and then you'll load the Promethean flow cell the same way as well, so with 50 to 100 femtomoles of cDNA. And then as you're starting this run, the MinKNOW parameters are the same as well for the 3' and 5' protocols. You'll be selecting the ligation 114 sequencing kit. You'll set your runtime to about 72 to 100 hours. I've had some runs go quite a bit longer recently, so you can definitely bump that up. And then you can either base call live with Dorado or base call post-run with Dorado, so that's up to you. So here's an example of what you should be expecting from a 3' and 5' protocol. This example here is shown with 5,000 PBMCs at 95% viability. So with the 3' protocol, you'll be expecting about 130 million total reads off one Promethean flow cell. And then after filtering and alignment, you should be expecting about 105 million cell-assigned reads. And then the read density of the 3' protocol is approximately 900 base pairs. So with the 5' protocol, you'll be expecting about 90 million total reads off a Promethean flow cell. And then after filtering and alignment, you'll be getting about 60 million cell-assigned reads with the 5' protocol. And the N50 of these are approximately 1,000 base pairs long. So I'd like to highlight this specific biological example. So these are B cells that were collected following a measles, mumps, and rubella vaccination. So this was a population of cells that was activated to this specific immune challenge. So on the UMAP to the left here, we're showing single gene analysis clustered by isoform expression. So you're now seeing isoforms that were different or preferentially expressed in some clusters, while other isoforms are being favored in other clusters. So here we have a BCL2 gene, which is critical for the apoptosis pathway. So you can see that the total expression of the BCL2 gene in cluster 3 is overall higher than in cluster 8. And then if we look at the transcript usage, so we have 3 BCL2 transcripts that are being differentially used. So in cluster 3, the BCL2-201 transcript is about 69% of the gene expression, whereas in the cluster 3, we have about 4% of the BCL2-202 isoform expression. And then if you compare that to cluster 8, the BCL2-202 isoform is at about 32% expression, so there's quite a difference there. But if we look at the actual isoforms themselves, so the BCL2-201 and BCL2-202 isoform, they express the same protein coding sequence, but they vary in the sequence of their 5' UTR. And then if we look at the third transcript, the BCL2-203, it's expressed as a single exon. But you do see a difference in expression between cluster 3 and cluster 8. So this is just an example of how single cell can add more information to your biological question. This would have otherwise been obscured with just gene level analysis. So the really beautiful thing about the Promethean line is that it's very scalable for your needs. So if you're working with like smaller cell types, just interested in cell clustering or transcriptome profiling, the P2 device can run 2 Promethean flow cells in parallel, and it's less than $11,000 to get started. So this would really pair beautifully with your Chromium device. However, if you're a core lab or a high throughput lab where you're working with millions of cells and billions of cell-assigned reads, we have a higher throughput Promethean device, the P24 or the P48, where you can run 24 to 48 flow cells in parallel. So this is going to be really great for atlas scale mapping, where you're looking at SNP calling and possibly low abundance isoform characterization. So the throughput of the Promethean is really scalable to your needs, so whatever application you're working with in the biological question that you're asking. So just in summary today, we've talked about how nanopore full-length sequencing of transcripts can help characterize isoforms, fusions, and you can also genotype across the full transcripts. We're compatible with the current 10x genomics platforms, as well as any cDNA that you might have laying around in your freezer. It's a 3-hour library prep. It's single tube, so it's really easy to prep more than one at a time. And we're flexible for the scalability, so you can run 1 to 48 flow cells depending on your device. It's also really easy because you don't have to batch them per Promethean flow cell. It's 1 sample per flow cell, so it's really nice and easy. I'd just like to highlight a talk later today at 4:10 in the Data Cafe, hosted by Matt Parker and Brian Leland. They're going to walk through the workflow single cell hosted on the EPI2ME platform. It's really easy to use, and it puts the analysis right in your hands. So that's all I have. Thank you.